So we ended the last portion of chapter 8 talking about transcription and translation. One thing interesting is that in prokaryotes, translation actually begins while transcription is still happening. In other words, transcription and translation can happen simultaneously or at the same time. Now, the fact that neither process actually occurs within an organelle is what makes this possible. Both transcription and translation are happening out in the cytoplasm of the bacterial cell, not with one happening inside of a nucleus and the other happening outside of the nucleus at a ribosome. So because the ribosomes and the DNA are actually in the same place, transcription and translation can happen together. In fact, translation can begin before transcription even finishes. It's also possible for bacteria to build what we call polyribosomal complexes. Okay? Basically, where several ribosomes actually attach to an individual piece of mRNA, and those ribosomes grab onto the mRNA and are all responsible for translating the same piece of mRNA at the same time. They're just at different positions on that piece of mRNA. This is only possible where transcription and translation both happen together within the cytoplasm, so it only happens in the prokaryotes. You can see it here. The blue strand, the blue strand is your DNA. These red strands are actually RNA that's being made from that particular piece of DNA. So the RNA is actually being constructed here. So this is an RNA polymerase moving down. Okay. So you see it's smaller here. So RNA polymerase is moving down the strand of DNA. As this happens, we have multiple ribosomes actually attached. Okay. So multiple ribosomes attached to the DNA, and each of those ribosomes is responsible for actually stringing multiple amino acids together to make a polypeptide. Now, some other differences between eukaryotic and prokaryotic transcription and translation. The start codon for both of these is AUG. Okay? But in eukaryotes, it codes for an actual different form of methionine. They're both still methionine. There's just a difference between prokaryotic methionine and eukaryotic methionine. Eukaryotic mRNAs code for a single protein, unlike bacterial mRNAs that actually often contain information from several genes in a series. Okay? We call these concerted genes. Basically what happens here is that multiple genes, if multiple genes are necessary to do a specific job, in other words, you need 2 or 3 proteins in order to break something down or build something, the thought behind this is that why not just make them all at the same time? If you need all 3 of them, why make one at a time and having this one gene, one protein rule? Okay. Transcription in eukaryotes takes place in the nucleus, where the RNA actually has to pass through nuclear pores out to the cytoplasm in order for translation to happen. Again, we just saw that does not happen in prokaryotes. Okay. The prokaryotes, everything is out in the cytoplasm. There is no passage through a membrane. Okay. And eukaryotic genes do not exist as uninterrupted series of codons. Okay. In fact, eukaryotic genes actually have interrupting sequences, okay, that we call introns. They're actually sort of sequences that are stuck in the gene that are never actually meant to be used to make a protein. Now, for eukaryotes, introns will actually be removed within the nucleus. So introns are removed in the nucleus, they stay in the nucleus. The exons are then joined back together. We call this splicing. And those exons get joined back together. Right? And that is what makes your complete piece of mRNA that will be used to translate a protein. We're skipping this one. This video is readily available. If you want to see it, it goes through everything that I just went through. Visually, it's available on cont'd. Okay? in the library tab in chapter 8 under the animations or videos section. So, we're going to talk a little about regulating protein synthesis, and honestly, a lot of the information we know today is information that we derive from bacteria like E. coli. Okay? We do know that we would like to induce and repress the expression of certain proteins. Okay? In other words, this is controlling gene expression again. Okay? Very similar to what we did in chapter 6. Okay? Except this time we're going to talk about mechanisms. Instead of just talking about inducing and repressing genes, we're going to talk about how it happens. Okay? Now, again, the reason behind this is that it conserves energy. Okay? Because making genes, or I'm sorry, turning genes into proteins, so making proteins is very energetically expensive. It costs a lot of ATP. So if you don't need it, there is no reason to spend the ATP creating something, specifically creating a protein, that's not going to be used by the cell. Okay? Now, what we're going to talk about is regulating a set of genes. Okay? A coordinated set of genes, in other words, a regulated set of genes called an operon. And operons are only found in bacteria. Okay? Here, this is a set of genes. The whole thing is regulated as a single unit. So either you make all of the genes in the operon, or you don't make any of the genes in the operon. Okay? They can be inducible or repressible. Okay? It depends upon the operon. In other words, they can normally be on and then turned off. That's a repressible operon. Or they can normally be off and have the capacity to turn them on. That's an inducible operon. So turning on, induction; turning off, repression. Lots of operons actually are encoding enzymes that are specific to certain catabolic processes. Okay? Usually it's a set of enzymes you need to digest or metabolize a certain substance. Okay? And it's really common for those enzymes to be turned on, okay, by the presence of that substance. If you remember, our example was glucose and lactose, except we called it Pop-Tarts and toaster strudels. We said that the only reason you were ever going to turn on that strudulase, okay, is because you're trying to digest toaster strudels. So the presence of toaster strudels should induce the production of strudulase, and the presence of Pop-Tarts should induce the production of tartase. Okay? So these enzymes that are needed end up being enzymes that are needed to metabolize the nutrient end up only being present when the nutrient is there. That prevents the cell from wasting energy making an enzyme that it does not need. So the specific operon that we're going to talk about is what we call lactose operon. Okay? Now, this was first described in 1961. It's actually three scientists, Jacob, Monod, and Pardee. Pardee doesn't get quite as much credit. Okay? The lac operon is specific for lactose metabolism. We're trying to digest lactose. So the only reason to actually make these genes that digest lactose is if lactose is in the cell. So the rest of the time, the gene needs to be turned off. Okay? So, three major parts of the lac operon. Okay? A regulator or gene that codes, okay, for a protein that's capable of repressing the operon. So we're going to have a regulator gene. Okay? It gets turned into a regulator protein. Okay? Downstream, there will be what we call a control locus. Okay? Two parts of the control locus: the promoter and the operator site. Okay? And then the actual, I should keep these connected. Okay? Structural gene. This is the, hey, here's how to make the enzymes that you're trying to make. And of course, eventually there will be a terminator region. If you remember genes looking like this. Okay? So let's really quickly, okay. Let's go to the white screen really quickly and redraw this. So, this regulator site, interesting thing about the regulator site, it's almost always on. So this will make regulator mRNA. Okay. It will make a regulator protein. Okay. I'm going to say the regulator protein looks like this. Okay. And then slightly downstream, okay, okay, we will find the promoter. Okay. Hopefully you remember the promoter is where RNA polymerase binds. Okay. The operator. Okay. This is a set of genes. Yeah, the operon is a set of genes. Specifically, the lac operon is a set of 3 genes. So gene 1, gene 2, gene 3, right, and then a terminator site. Okay. This is what we're finding on the DNA. Okay. Now, the issue here, okay, and the thing that sort of keeps us going, okay, is the presence of, okay, this regulatory protein. Okay. The regulatory protein is what's going to be in charge of doing all of the control of whether or not we actually get to make this gene. Okay. So, if you recall, RNA polymerase sits itself down at the promoter. Okay, and moves down the gene. Okay. Making RNA. Basically making a strand of mRNA that we can send to a ribosome and we can use it to make proteins. Okay? But it's got to be able to move all the way down the DNA. Okay? Well, the thing that regulates whether or not this can move down the DNA is the presence of this regulatory protein. So what happens is the regulatory protein, okay, actually sits itself down, sort of straddles this operator site. Okay? And while that regulatory protein is attached to the operator site, okay, it acts like a roadblock. Okay? It prevents the RNA polymerase from moving down the DNA and using it as a template to make RNA. Okay? No RNA means no protein. Okay? Specifically in this case, no enzymes. You're not going to make this enzyme, or this enzyme. Okay? They're going to get canceled out. Nope. So that regulatory protein is what is regulating whether or not this gene can get used. Now, kind of the interesting thing about the regulatory protein is that the regulatory protein can be removed. Okay? The regulatory protein actually has a much higher fondness for lactose molecules. So this is lactose. Okay? When lactose is in the cell, we need these enzymes to digest lactose to use it as a nutrient. Okay? So when the lactose comes into the cell, what happens is the lactose bonds to the repressor protein and removes it. Okay? The lactose bonds to the repressor protein and moves it. It takes away the roadblock here, okay, and allows us to make mRNA, which gets us, okay, protein 1, protein 2, and protein 3, those enzymes that you need for lactose digestion. Okay. So we can see it here. Okay. Hold on one second. I'm going to move on to a picture in just 1 minute. A little bit more about the lactose operon. Gives us an efficient strategy so that genes can actually utilize a specific metabolic pathway, okay, and gives us the ability to induce or repress that particular pathway. It functions only when lactose concentrations are high in the cell. In other words, the glucose is gone because the glucose is the food of choice. Okay? In other words, we've eaten all the Pop-Tarts and had to switch over to toaster strudels. So when lactose is brought into the cell as an alternative form of sugar, okay, in other words, the glucose is gone, we just switch over to lactose metabolism. Okay. Basically making sure that we can produce those 3 lactose digesting proteins. Okay. You can see it here, right? This is our regulator. Okay. The P site is the promoter site, the O site, the operator site. Okay. That repressor protein moving up here. Okay. This means no mRNA because that RNA polymerase can't move downstream. Okay. Okay. And that means no proteins. Okay. No enzymes. Okay. But if lactose is present, okay, if lactose comes in and binds to the repressor protein, it inactivates the repressor. When we take away the repressor, we allow, okay, our RNA polymerase, this guy right here, to have moved all the way down the gene, okay, to produce mRNA, and for translation to produce the 3 enzymes necessary to digest lactose: beta-galactosidase, lactose permease, and lactose transacetylase. Okay. Now, when lactose is used up in the cell, the lactose concentration goes back down. Okay, that repressor protein will not be inhibited by lactose, and it can move itself right back up, okay, to sit at the operator site. Again, there is a video of this, okay, an animation found in your textbook, okay, on the website, specifically in the library, in Connect Chapter 8. All right, we're going to skip out on recombinants a little bit. Okay. But I do want to talk about plasmids. Okay. The reality is when we talk about recombinants, the simple definition is where one bacterium gives another bacteria DNA. So getting DNA from a different source makes an organism a recombinant. Okay. Now, one of the easiest ways to do this is by attaining a plasmid. Okay. And if you remember, plasmids are single small circular pieces of DNA. Our buzzword for plasmid is extra chromosomal. Okay. If you see that anywhere in a test question, okay, your brain should automatically go, oh, we're talking about plasmids. Okay. When you look and see plasmids is in the answer anywhere, we're talking about plasmids. Okay. Extra chromosomal equals plasmids. Okay. All right. Now, the deal with plasmids is that they are used to pass useful traits from one organism to the next. Okay. This is a type of what we call horizontal gene transfer. Okay. Any transfer of DNA that results in an organism acquiring new genes that didn't come directly from a parent. In other words, acquiring DNA from another organism or outside of the cell, anything that's not provided via binary fission or cell division is considered horizontal gene transfer. Now, this comes in 3 types, 3 mechanisms for horizontal gene transfer. conjugation, which you're actually already quite familiar with, where we pass a plasmid through a pilus. That's always how I try to remember it. Passing a plasmid through a pilus. Okay. Transformation, where cells actually uptake naked DNA. In other words, DNA that is not inside of a cell. Realistically, this is DNA that's kind of floating around in the environment. So transformation is the uptake of naked DNA. So this is very commonly done in the lab. Honestly, it's rare in nature. Okay. It does happen, but it's a lot more rare than things like conjugation or even transduction. Okay. In transduction, we see DNA carried from one bacteria to another via a virus. Okay. So there are all kind of keywords that go along with this. Passing the plasmid through a pillid. This is the only mechanism that requires contact between two organisms. Transformation, that naked DNA. Transduction, anything that has to do with a virus. So when the virus passes the DNA from one cell to the next, that's considered transduction. Okay. All of these are what we consider horizontal gene transfer. Okay. So conjugation, again, the conjugation pilus, you're used to seeing this. Okay. All right. Where one organism is actually moving information, specifically a plasmid, extra chromosomal DNA, from one bacteria to the next. Okay. Commonly moved, okay, genes that you can find on plasmids. So these are biomedically important genes that you can find on plasmids. Okay. F plasmids or fertility plasmids have the genes necessary for conjugation. In other words, these are the ones that make conjugation possible. If you remember, we also called it an F pilus or a sex pilus or a conjugation pilus. Okay, so having this makes an organism able to move a plasmid from itself to another organism. Okay. Resistance plasmids, these are going to be dangerous. Okay. Basically, the genes on a resistance plasmid are going to confer resistance against an antibiotic or possibly some type of cellular poison. The bad part about resistance plasmids is one thing we found is that usually they confer resistance to sets of antibiotics, not just one in particular. So it's not just being resistant to penicillin, it's being resistant to all the penicillin-like organisms, or all the ones that have beta-lactams. Okay. Col plasmids, which contain genes that code for determining the production of bacteriocins. This has been one of the most commonly missed test questions. It has been on the last two tests. Okay. Bacteriocins, having to do with microbial antagonism. Okay. Where organisms will help keep other organisms out. Okay. So those good bacteria are taking up space and not letting those bad bacteria in. The bacteriocins, I said, were like mace, basically used to poison invading microorganisms. Okay. Degradative. Okay, sorry about that. Degradative plasmids that enable organisms to digest unusual substances. Okay. And virulence plasmids, which actually increase the virulence of the organisms themselves, usually by adding on a virulence factor, i.e. the ability to make pili, being more sticky, creating a toxin, creating an additional component within the slime layer. Virulence plasmids add on a virulence factor. Right. Transformation, basically where DNA from a dead cell is sort of released by the cell, usually when the cell dies, and nearby similar cells actually uptake the naked DNA fragments and then integrate those fragments into their chromosomes. Okay? Cells that are capable of actually accepting these pieces of DNA are what we call competent cells. This is much more common in the lab. Okay? If you remember, we talked about genetically engineering cells like bacteria or yeast to make insulin. Well, this is how we do it. We take a human insulin gene, okay, insert it into the cell, okay, and then get that human insulin gene integrated into the chromosome of the cell, and it's there forever. Okay? The cell has to be competent to take up the gene, but once it's there, the cell will produce that particular protein or enzyme. Okay? Now, in transduction, what we see happening is that a virus invades. Okay? A virus invades a nearby cell. Okay? In this case, it's a bacterial cell. The virus invades the cell, chops up the cell genome in order to make new virus pieces. In the process, one of the viruses accidentally picks up some leftover bacterial DNA. In other words, this is this right here. And when he moves on to a new bacteria, instead of carrying virus DNA with him, what this virus brings in is DNA from a bacteria, and that bacteria can be integrated into the new bacterial cell's chromosome. This is the end of chapter 8. We are stopping here. More on viruses in chapter 5.